This program is presented by University of California Television. Like what you learn? Help others discover UCTV podcasts by leaving a comment or rating for us in iTunes.
Our next speaker, Minnie Sarwal, has been a good friend for a long, long time, and most recently, a colleague, as she has joined UCSF in the Department of Surgery. Minnie has taken a long road to come to UCSF. She was a professor of pediatrics at Stanford, and then headed the personalized medicine lab at Sutter Hospital. However, as you can imagine, she didn't have to travel far to come to UCSF. Her topic today is, I think, one of the big buzzwords in medicine, and one that we haven't yet fully incorporated in the transplantation, and that's personalized medicine using gene-based assay. Minnie?
Thank you so much. It's very exciting and a privilege for me to be actually part of this amazing transplant program at UCSF. And my lab is also very excited. We have a beautiful lab on the 12th floor, one of the buildings at UCSF with a huge expanse overlooking the Bay Area, which is very exciting for us. So I'm going to talk about genomic monitoring for transplant patients with the idea that I think the talk previously was very exciting about the future of trying to really replace, I think, the need for transplantation and to really improve on really the quality of life of patients with end-stage renal disease. I'm going to focus more on how can we better monitor the patients once they've actually had an organ transplant, given the fact that we have the huge problem with organ shortage, as well as the really finite life expectancy of allografts. We've done a lot of work with different solid organ transplants, really addressing this approach in kidneys, heart, lung, as well as liver. We've looked at the axis of trying to monitor transplant patients by really taking an approach of saying, we really don't know what is happening. If you look at the literature, there are lots of genes and proteins and molecules that people have studied that have associative kind of findings as well as correlations with perhaps this T-cell related gene goes up when you have rejection, or perhaps certain genes associated with Tregs go up if you have long-term graft acceptance. But we still haven't been able to develop an assay that really performs with a high level of sensitivity and specificity that we're going to need as clinicians to apply these tests to actually change practice for patients. So as a clinician, if I look at these assays and I say, am I going to use some markers that are coming out of certain studies and actually start changing treatment for my patients? I think there are two issues. You're going to need an assay that has sensitivity that's going to be pretty high, perhaps greater than 80 to 85%, given the fact that the sensitivity of the creatinine, at least for diagnosis of acute rejection, is pretty good. But then I think there is also the issue of specificity of an assay. The specificity of the serum creatinine for diagnosing rejection is about 20%. But I think we need assays with very, very high specificity. We need to know that the patient is safe so that we can actually let them go and not get them back for intensified treatment. And so I would say that those assay specificities have to be greater than 90%. So a very high bar really going into doing any kind of mechanistic studies and applying them to patients. So I'm going to take you just through the genomic axis of some of the work that we've done, focusing really on kidney as well as its application to heart. And so there's first an introduction of how we've done some of these studies, and then really ending by taking what we've learned and taking this to a panel of genes which we think can be applied to actually manage patients. So the omic approaches, or omic is basically genomics, proteomics, antibiomics, basically large-scale interrogation of big data sets relating to genes, proteins, et cetera. And of course, you have genomics, where you can do sequencing of genes. There's transcriptomics, where you can look at the mRNA level. There's proteomics, where you can look at the proteins. And then you can look at the interaction of all of these really pathways with each other to better understand disease mechanisms. So, but when you take these tools and you apply them towards monitoring, what are we really trying to do for our patients? We're really trying to understand, I think, a few things. We want to understand baseline risk, and I think we heard some talks of how clinical criteria are put together to optimize management of patients for the clinician, looking at various demographics, looking at various clinical parameters, sensitization status. So can we take those kind of genomic or proteomic studies and actually better stratify baseline risk? So I think for that, probably genotype studies and sequencing studies, which are coming in, I think would be very important, and I'm not going to talk about that right now. You have the initiating event, which is transplantation, and then you actually basically have an HLA mismatched organ that is actually engrafted, and then you have a host of different alloimmune responses against the graft, which can escape even in the presence of lifelong maintenance immunosuppression. So you have a preclinical process which we would like to capture to really understand, can we predict some of these events really prior to the clinical manifestation. And for this, basically, as I said, there are SNPs, there are mRNAs, there are proteins, et cetera. But really, the reason we do this is because we want to really try and predict which patient has the highest likelihood of rejection and conversely tolerance, and which patient would be perhaps best suited to which particular immunosuppression cocktail. And in the post-transplant state, you really want to look at the risk of rejection, try and figure out which patients perhaps are accepting their graft such that you can minimize immunosuppression prospectively, and then really use these assays to predict graft outcomes. So these are all, I think, lofty ideas. How do we actually put this into practice? So the way we've done this is we've actually spent the last, I would say, 17 years actually doing this, where we've actually collected transplant patient biomaterial. We have freezers in our labs now that have over 20,000 human samples, thanks to the efforts of the patients, the clinicians, et cetera, that have actually collaborated with us over these many years. So these are all the different kind of tissue samples that we've collected from these patients, and these have been matched with very carefully collected demographic data on these patients. We've then applied this to high-throughput discovery, saying that we don't believe that just looking at, say, granulysin, granzyme, FAS is just a marker of acute rejection. There is a lot that we don't know. So we put this through high-throughput discovery methods, as I said, looking at various platforms. We then basically develop marker sets of genes and proteins that we validate through a particularly well-annotated, collected set of samples from patients that are very, very well studied. These are put through various bioinformatic analyses. And then it's very important, if you get a set of genes that says these track with rejection, say, there could be a lot of other confounders that exist within your data set that you haven't fully identified. So you have to take these genes to a completely independent data set, ideally even demographically different, that you can then validate and cross-validate really the performance of those gene sets for really the prediction of the same clinical phenotype. And here we're going to be focusing for purposes of really, in the interest of time, on rejection today. And then there's this last part that I have really had the privilege of getting to know how to do this over the last 2 years. As an academic person, it's very exciting for us to actually ask these questions, generate mechanisms, generate biomarker panels, publish 25 to 30 papers a year, publish in high-impact journals, go and give lots of talks. But have we actually really translated this to clinical practice? Have we actually brought these tests to patients? And if you actually look at our field, is very difficult. And the problem is here, because actually taking that assay and taking it and reducing it to a test that can be run in a CLIA certified laboratory, can be offered as a clinical test for which you can get reimbursement from insurance, it's a very long road, and it actually takes millions of dollars and a lot of work to actually get that downstream. There's a whole process of regulation, and I think we heard about how the involvement of FDA into translation of some of these tests is very important. So this is, I think, as we're moving into the era of personalized medicine, it behooves us to really understand how complicated, expensive, as well as time-consuming this process is to actually apply these tests to patients. So I'm not going to focus so much on really the sequencing end of it, but I think this is, again, really in our future. Not just in our future, it's actually here today for a lot of tests. The genomics is understanding the sequencing of genes, coding for functional proteins and regulatory motifs, understanding the individual differences in genetic composition, doing the studies involving next-generation sequencing are here. And the reason is because we all want to do personalized genomics. We want to use these tests to discover genomic variation. Why am I different from the other person? What is my risk of hypertension? What is somebody's risk of breast cancer? So you want to basically develop these genotype-phenotype associations, and some of these assays have already been commercialized. So we've seen the path forward. You have 23andMe, where you have a buccal swab, you get it back, and well, you can be argued that is that data really interpreted to the degree as well as the amount of density that should be done, but still, these are tests that are available that can start to give people risk susceptibilities of various diseases. I think it's very important for us to bring these assays into transplant, but we basically have a domain which is incredibly complicated by the comorbidities that exist in transplant recipients. So I don't think this is going to be immediately in our future just because of the vast amount of work that has to be done, especially bringing this to transplant patients. So I'm going to focus more on the next step, which is really looking at mRNA levels rather than sequencing, so rather than looking at DNA coming to RNA. And so you can look at differences in regulation of RNA in different biofluids. You can look in the organ, you can look in the blood, you can look at the urine. And I'm going to focus, even though we've done these studies in different tissues, I'm going to focus today just on blood. So I'm going to show you how you can harness differences in RNA expression in blood to actually better understand and non-invasively diagnose the acute rejection process, but perhaps also predict it. So the reason to do this is because we know that we all face the problem of drug dosing, and we want to do it so that we can minimize rejection, but also minimize infection, malignancy, as well as all the comorbidities of chronic immunosuppression. So the reason is we want to be able to dose immunosuppression to the immunological threshold of the patient. It's pretty much like telling somebody who's managing a hypercoagulable patient that just give them warfarin, and you don't have any parameter to measure really what their dosing should be for this drug, and you should wait until they clot, which is essentially rejection, the equivalent of rejection, or you should wait until they bleed, which is perhaps over immunosuppression of cancer. That's not how we dose anticoagulation. But that is how we're actually essentially dosing transplant patients. So it's very important that we actually determine the immunological threshold for each patient such that we can better dose immunosuppression. And the reason this is so important is because these patients, this is just showing, a standard patient and the amount of drugs that they have to take. Probably 2 or 3 of them are actually their immunosuppressive drugs. The remainder of the drugs are actually to manage the comorbidities, largely driven through the usage of these drugs. So I'm going to show you that the development of an assay, as I said, the assay needs to have a very high bar to be accepted, but the assay also needs to be able to be non-invasive, so it should be able to give you the information about acute rejection without the need of a biopsy, and it also should be done so that it can be predictive prior to perhaps clinical graft dysfunction or a rise in the serum creatinine. And we know that currently there is really no reliable blood test to diagnose or predict rejection or tolerance. And this is basically a laundry list of why it is so difficult and why, even though there are so many labs all over the world working on it, why haven't we been more successful? Because there are all these confounders that exist in patients, and I'm not going to read through these. So to do this, I'm going to take you through how we have actually and this has taken us about 16 years of work, to actually develop a gene-based blood test to optimize patients. And this is basically to really monitor non-invasively in blood for diagnosis as well as prediction of acute rejection. And these genes that we have finally been able to find actually work for diagnosis not just of kidney, but also for heart rejection, suggesting that we have actually honed into the mechanism of looking at differences in RNA expression in blood that actually exist across all different solid organs. So we've done this really by taking microarrays. This is a powerful means to quantify the expression of the entire genome in a tissue. So it's an a priori kind of, I really don't know what I'm looking for, but I'm going to go in and find the differences that are most biologically significant. So we've taken blood tests. Each blood sample has been paired with a biopsy. These blood samples have then been processed on microarrays, and basically you get these red-green kind of beautiful signals. So red means that the gene is up in that sample. Green means it is down in that sample. So it's basically a relative comparison. And we've done basically lots of elaborate bioinformatic tools. We've published basically a methodology that we published recently in Nature Methods that allows us to deconvolute where the signals of these genes are coming from. Because when you take a whole blood sample and you process it, it consists of lymphocytes and neutrophils and monocytes. And so where is this signal coming from? And in the interest of time, I can't go into the details of this, but there is a way that you can bioinformatically deconvolute where the signal is coming up. And we found that the signal of the genes that traffics with the diagnosis of rejection in the graft actually comes from trafficking monocytes, which are actually in the periphery. And because it's trafficking monocytes, we believe that's why the signal is common, irrespective of what kind of solid organ is being transplanted. So this is a multi-parameter approach. We actually use 3 different kind of microarray platforms. The Affymetrix, Agilent, Lymphochip, these are all different names, but they are basically cDNA-based or oligo-based platforms. I've just put the slide up to show the amount of work that goes into trying to discover a handful of genes. It's the true looking for the needle in the haystack approach. And I think it's, I have to say, had I standing here, would I have still done this kind of research? If I could look back 16 years, probably not. It was probably the most expensive publication that anybody has done in science, that you wait 16 years, you spend close to $16 million, and you have one paper out of it. So, but regardless, I think it's the belief and the passion that we needed to make the difference that actually kept us going. So we basically did microanalysis. We came up with a set of genes. We controlled them for different clinical confounders. We sorted cells from patients and did arrays of them. We discovered some genes, we verified them, we selected them and built models. We validated this in a multi-center clinical trial that was run through the National Institutes of Health, initially in pediatric and young adults, and then subsequently in adults. So what is the data? So this is the simple test. So it is truly a needle in a haystack. From those 43 genes, we found that we could actually develop a logistic regression model using as small as 5 genes in this multi-center trial. That model was locked, the genes were assayed blindly in the lab, and all of the analysis was done by statisticians at the NIH. And the results were this y-axis that you can see here from 0 to 100% is basically the predicted probability of acute rejection based on the logistic regression model of these genes, and the names of the genes are shown up there. These are all genes that have not been previously ever found to be, at least on single gene basis, to be studied in acute rejection. And you can see in this logistic regression model, the red dot is a blood sample that is matched with a biopsy that had confirmed histological rejection. And the green dot is a blood sample that is taken with a biopsy that showed complete absence of acute rejection. All the biopsies that came from this trial were centrally scored by Dr. Richard Sibley at Stanford, and so everybody was blinded to really the data. And you can see the logistic regression score actually separates the patients pretty nicely, almost into kind of a binary end. So you can see most of the acute rejection kind of blood samples have scores which are like greater than 80%. And most of the green dots are people who actually had stable grafts in the absence of acute rejection. You can see that these dots are pretty close to like 0 to 20%. So that's great. You want an assay to be really at the two ends of the spectrum. You don't want the assay to be 48% is no rejection and 52% is rejection. I think that would be a very tough assay to really work in clinical practice. You can see that there is some scatter around the center, but that scatter is quite little. And so if you look at the sensitivity and specificity from this multi-center clinical trial, 120 patients basically in that trial, actually 130 in the trial, 120 in this assay, you can see the sensitivity and specificity right there, 99 and 97% is very, very high. So this study was done in pediatric and young adults. Since then, I'm just going to show you a couple of slides, which is unpublished data. This paper is currently under review. We've gone on to amass 527 unique renal transplant adult and pediatric patients. These have come from the following centers. You will recognize the names of these are some of the leading transplant programs in the US. We also were lucky enough to have samples from Barcelona. We have one of the professors who actually sent us samples from Barcelona sitting in the lab, sitting in the audience, who's actually in my lab for a year here at UCSF, as well as samples from the University of Mexico. So you can see there were different blood samples that came from these different centers. They were picked up by the principal investigators at these sites, and they came here for testing against against the genes that we had. I'm not going to go through this whole process, but suffice to say advances in technology have really helped. So we were lucky enough from the NIH to be funded to get the Fluidigm Biomark machine. This is a humongous kind of behemoth monster-looking machine. But you can basically, in one day, get 9,000 PCR reactions, which on the standard ABI platform would take us 8 to 10 weeks to get. So you can see that advances allowed us to really rapidly scan all of those 43 genes across all of these huge number of blood samples to really optimize the selection of genes that would work in adults as well as pediatrics, irrespective of different induction protocols, immunosuppression protocols, as well as comorbidities. And this basically shows just one of the slides from the paper, and you can see at the bottom are basically the mean and standard deviations of the scores, again shown on the y-axis there, 0 to 100%. Of when you have groups of patients, again in red is basically with rejection, so blood samples with the scores, when you had matched biopsy-proven rejection, and green blood samples taken from the patients who actually did not have any acute rejection, again blinded to the results when the studies were actually done. So you can see this was very exciting, and these are the PIs that have actually contributed to some of these studies. So more importantly, I had talked about diagnosis. It's very exciting. We have an assay that really can quite accurately mirror what is happening in the graft at the time of rejection. But can you predict this? So if you look at stable people and you look at them over time post-transplant, you can see that these scores of the genes, they are not really affected by time post-transplant. Because one question could be is that when you have hefty immunosuppression in the early post-transplant period and low immunosuppression later on, do these scores really change? And you can see they don't. More importantly, can these scores predict? And I think this is data that we had in a subset of patients when blood samples were actually collected prior to an acute rejection episode because of protocol sampling. So you can see if a blood sample was collected and analyzed within a 3-month window prior to an acute rejection episode, or a 6-month window prior to an acute rejection episode, and you looked at those scores, they were dramatically higher. These p-values were less than 0.001 when you compared these scores compared to people who never had acute rejection episodes. So that told you that two things, that you could actually start to tell which people were going to develop acute rejection, because in these two cases, these patients had stable creatinines. The clinician was following them. They were at home. They hadn't come to clinic. There was no way to tell that they were actually headed for a rejection episode. So I think the first thing is you can start to predict who's going to have a rejection episode prior to significant injury to the graft such that the serum creatinine rises. And the second thing is you could perhaps monitor this serially. And you could see if, in fact, these scores were rising over time, suggesting that the risk for rejection is increasing. And you can see when these people were actually treated by intensification of immunosuppression by the physicians, whether it was solumedrol, thymoglobulin, whatever, you can see that there is a decrement of these scores when we follow the expression of these genes over time. So it's important to understand that actually these genes do not separate between antibody-mediated rejection and cellular rejection, as well as also if the rejection is early versus late post-transplant, these scores don't actually seem to discriminate for that, which is similar to what we saw with the stable patients. And you can see that this continues, this is the same, so you can see essentially that this is just repeated here, apologize. So you can see the application of these kidney rejection biomarker genes, since they came from trafficking monocytes. One of the questions was, would it work in heart transplantation? And Dr. Hannah Valentine, actually at Stanford, had in her freezer archived blood samples that had been matched with endomyocardial biopsies as part of a multicenter cargo study. And they were actually given to us, and we basically know that non-invasive methods are very important in heart transplantation. So basically, the best set of genes from the 43, the 10 gene set, was actually run in heart transplant patients. So if you see, this is a complicated slide, but I think you can see at the bottom here, we ran the PCR and developed a similar logistic regression model on these heart patients, and here is the data. You can see that, again, these are the scores, 0 to 100%. Here they're not in, the bars just show the scores, and you can see the phenotype is on top. So presence of CMV infection, which is a big confounder in heart transplantation, does not impact the scores. Stable patients, you can see there are a few outliers, but overall these are quiescent. And you can see most of the heart transplant patients where blood samples were collected with a biopsy with different grades of rejection actually had very high scores. Very importantly, all of the acute rejection 1B grades in heart transplant, which are currently not treated, all had very, very high scores. And I believe, I'm not a cardiologist, but I believe that in fact there is other literature suggesting that grade 1B rejections in heart transplant patients could in fact be in fact quite important to treat and could be pathologically relevant. And this basically shows the scores basically that spread across the different rejection grades. Again, if you look at these heart transplant patients who were stable, you can see these scores in people who never have rejections are quite quiescent. Again, you can see the same kind is seen if you have at rejection, your scores are high. Within a 6-month window, you can see that these patients, the arrow bars are slightly bigger, but you do have, in fact, elevation of these scores prior to detection of endomyocardial biopsy-proven rejection in these patients. So I think in summary, for the heart part, it looks like gene-based classifiers can be developed that work in heart transplantation. And in fact, there is a commercially available assay called AlloMap, which maybe some of you have used. Unfortunately, that assay has very high specificity, but only has 4% sensitivity and is impacted by time post-transplantation. So I think we do need to have additional assays that come in and do better for our patients by improving sensitivity and also not having the confounder of time post-transplantation. So I'd like to end by saying that the current monitoring of graft function, I know I would say relies on very dated technologies. I think the serum creatinine, yes, I know it costs whatever, $1, but I think really the information that we get back from it is also, it's a great surveillance tool. I think it tells us the warning signs, but these warning signs are actually pretty late. So I think these kind of technologies and the application and reducing them to smaller sets of genes have actually allowed us to identify sensitive and specific markers for rejection. Very importantly, 16 years of work was needed because we needed to find the genes that are not impacted by other multiple external factors that our transplant patients face. It's very important that these gene sets come from active trafficking monocytes based on some of the other work and informatics that we have done. And it's important that these genes are common in heart as well as kidney transplantation. This assay is called we've called it the solid organ rejection test or the SORT assay. It basically identifies humoral as well as cellular rejection. Very importantly, this assay is currently being validated in Europe in a 4-center trial in Sweden, where the assay is being done prospectively in about 220 patients. The assay is also being used in Mexico, where it's being used to actually stratify patients who will face immunosuppression intensification based on the readout. And just as Dr. Vincenti had shown, we were actually very lucky to be funded by the NIH for a grant, which will actually also start testing the same application in patients, kidney transplant patients at UCSF. And for anybody who's interested in looking to see if you can use them in your patient populations, the assay is being offered by Knight Diagnostics in Oregon, and that is their website. So I think I just want to end by saying that people showed lots of these different pictures of red, green kind of Christmas trees. There are lots of these heat maps that we can show and talk about, exciting publications. But really the time is not about heat maps anymore. It's really about translating this into tools that can be applied to patients. And this is actually a quote from the director of the NIH, Zerhouni, in 2005. And I think this is true even today, that at no other time has the need for a robust bidirectional information flow between basic and translational scientists been so necessary. So if you look at basically the public databases, this shows that there are 600,000 data sets that are actually available for all of us to analyze. And these are increasing exponentially every day. So you can see the number of assays at the bottom. It shows that there is 155,000 assays. So there is a lot of data out there that we can take with all of the sophisticated tools to try and understand a priori what these different mechanisms could be for better monitoring our transplant patients. And so I think I just want to end by saying, when we have these kind of powerful tools, how do we move forward into the next decade? I think we have to know what right question to ask. We don't just want to be taking these tools and building them and writing papers, but we want to really apply them to patients. We want to look at data sets, not just from our groups, but from groups worldwide, to better understand the biology of disease. And really, I know mice are great to study, and I know non-human primates are very essential to study for phase 1 trials and new drugs, but really the time is here to bring these tools and apply them to humans. So the era of personalized medicine is really here, and I'd encourage all of you to be involved with us and trust us to actually have some of these transplant patients at big centers like UCSF actually experience perhaps the dawn of that kind of era where personalized medicine will actually come to them, not just improve their quality of life, but also extend graft life expectancy. So I just want to end by thanking really people in my group. Some of them are in the audience, all the collaborators at Stanford, as well as all of the principal investigators at major transplant programs that have trusted us with patient samples, as well as the different funding institutions at the NIH, as well as industry. Thank you very much.
Long time, and most recently, a colleague, as she has joined UCSF in the Department of Surgery. Minnie has taken a long road to come to UCSF. She was a professor of pediatrics at Stanford, and then headed the personalized medicine lab at Sutter Hospital. However, as you can imagine, she didn't have to travel far to come to UCSF. Her topic today is, I think, one of the big buzzwords in medicine, and one that we haven't yet fully incorporated in the transplantation, and that's personalized medicine using
really replace, I think, the need for transplantation and to really improve on really the quality of life of patients with end-stage renal disease. I'm going to focus more on how can we better monitor the patients once they've actually had an organ transplant, given the fact that we have the huge problem with organ shortage, as well as the really finite life expectancy of allografts. We've done a lot of work with different solid organ transplants, really addressing this approach in kidneys, heart, lung, as well as liver. We've looked at the axis of trying to monitor transplant patients by really taking an approach of saying, we really don't know what is happening. If you look at the literature, there are lots of genes and proteins and molecules that people have studied.
This program is presented by University of California Television. Like what you learn, help others discover UCTV podcasts by leaving a comment or rating for us in iTunes.
Our next speaker, Minnie Sarwell, has been a good friend for a long gene-based assay. Minnie?
Thank you so much. It's very exciting and a privilege for me to be actually part of this amazing transplant program at UCSF. And my lab is also very excited. We have a beautiful lab on the 12th floor, one of the buildings at UCSF with a huge expanse overlooking the Bay Area, which is very exciting for us. So I'm going to talk about genomic monitoring for transplant patients with the idea that I think the talk previously was very exciting about the future of trying to that have associative kind of findings as well as correlations with perhaps this T-cell related gene goes up when you have rejection, or perhaps certain genes associated with Tregs go up if you have long-term graft acceptance. But we still haven't been able to develop an assay that really performs with a high level of sensitivity and specificity that we're going to need as clinicians to apply these tests to actually change practice for patients. So as a clinician, if I look at these assays and I say, am I going to use some markers that are coming out of certain studies and actually start changing treatment for my patients? I think there are two issues. You're going to need an assay that has sensitivity that's going to be pretty high, perhaps greater than 80 to 80.